Alright, and our last group of non-endospore making gram-positive bacilli is in the genus Mycobacterium. Now, why this genus might sound a little familiar. This is the bacterium, or bacteria, that makes the mycolic acid, that lipid in their cell wall. Now, although they are technically gram-positive bacteria, they will not gram stain properly because of that lipid in their outer cell wall. These are the ones that we have to do the acid fast stain. Now, because they're kind of their unique group, they have their own unique characteristics. One, they're slow to grow. So they're going to take a little bit longer to grow if we try to grow them up in incubators. Partly it takes a little extra time to make that mycolic acid. They do have protection from breakdown after phagocytosis, so they can be eaten, but then not broken down right away. They can grow then intracellular. Again, that's the best hiding place ever if you can live inside of cells. And they're starting to become resistant to many antimicrobial drugs. They're resistant to heat, to drying, to various chemicals and disinfectants, so they can be harder to get rid of. Now, there are over 75 species of Mycobacterium that can affect, or that are out there. However, there's only 3 that infect humans. So we're going to talk about those 3. We don't care about the other 72. And the main one that you might be the most familiar with is Mycobacterium tuberculosis. Also just shortened it to TB because it causes tuberculosis. It's a pulmonary disease that, yes, we get it when we breathe it in. There are lots and lots of cases out there. They are on the decrease, but a lot of individuals go undiagnosed for a long period of time. Now, there's 3 types or 3 stages of tuberculosis: primary, secondary, also called reactivated, and disseminated tuberculosis. Now, the next slide kind of shows the better pictures of these 3 stages. So that primary tuberculosis, this is a lung that actually have some of these tubercles growing in them, those white spots. But primary tuberculosis is once those bacteria get into the cell, or into the body, they will get ingested by a macrophage, a big eating cell. That's its job, is to eat bacteria, but they don't die, they don't get broken down. And our immune system's job is to try to take all those infected white blood cells and kind of clump them all together into this big, huge structure called a tubercle. It's where the tuberculosis gets its name. Now, the tuberculosis, these kind of confined, isolated, infected cells don't last forever. At some point, those cells are going to start to die, and they start to die kind of from the inside out. That's this caseous necrosis. Now, they say that this, death of these cells, and because it kind of almost forms a yellowish look to it, kind of looks like Swiss cheese, which is why this particular organism, yes, is holding some Swiss cheese, is that the actual tubercle, as it starts to break down, kind of has that consistency. Well, this is primary tuberculosis. We've got all these infected cells forming the tubercle, and yes, some of those tubercles can be dying. Now, it's when we get into the secondary reactivated tuberculosis is when that dying tubercle ruptures and that bacteria starts to spread to other alveoli in the lungs, and it can sometimes get into the bloodstream, but it is spreading. Now, if it's still in the lung area, it's still secondary tuberculosis. It's only when it gets into other parts of the body that we call it disseminated tuberculosis. So this is the pathogen traveling to other parts of the body. Apologize. And so a lot of places that this particular bacteria likes to get to once it gets into the bloodstream: the spleen, the kidneys, the brain tissue. Generally, by the time it gets to disseminate, it's usually only affecting those that have more of an immunocompromised immune system, or those, especially the HIV patients, that really have no working immune system. Now, diagnosing it, one, they can look for that acid-fast positive smear, usually done off of a sputum sample, again, looking for in those lung tissue. They can culture it out, they can grow it, they can look for the bacteria itself. They can do genetic probes looking for the DNA of the bacteria, or they could do a tuberculin skin test. Again, most of you had the TB skin test done, and I'm going to guess most of your results did not look like this. The idea for those skin tests is they inject the antigen for the bacteria. So they don't give you the bacteria, but they do give you parts of the antigens to this bacteria. And if you have antibodies against it, it means at some point you've been exposed to this bacteria. Doesn't mean you have an active infection, but at some point you've been exposed to this bacteria. You have antibodies that are going to combat and kind of fight off and cause this inflammation at that antigen site. They can also do chest x-rays looking specifically for those growths inside of the lung tissue. Now, treatment is hard. We do have a lot of antimicrobials, not a lot, we do have some antimicrobials out there, but most of them have to get used together in combinations and for months. Again, most antibiotics, 10 days, 2 weeks, these are hard to kill organisms. The top antibiotics that are most commonly used is like I'm going to always say these wrong, isoniazid, rifampin, ethambutol, and the pyrazimide. Yeah, you can read it. Well, there is a vaccine though, so we do have a vaccine on the market. I was going to say the BCG, named after the people that came up with it, and usually it's only administered in areas that are at high risk or if there's, a prevalence, an outbreak happening. Your best bet is to avoid inhaling droplets from someone that is infected with TB. And so wearing masks or having, the patients wear masks. If you're exposed to someone that has it, you should be wearing a mask so you don't inhale that bacteria from them. Another bacteria, not as common, that's in the Mycobacterium group, is the Mycobacterium leprae that causes leprosy. It is a bacteria that only can grow at 30 degrees Celsius. We haven't been able to create any plate that can grow this in the lab, but if we want to grow this, we grow these in armadillos because they have a body temperature of 30 degrees Celsius. Now, because they like and only grow at 30 degrees Celsius, the only places they really grow in the body are the cooler regions of the bodies, the extremities, the ears, the fingers, the toes. It is an extremely rare bacteria, and how it's transmitted is nasal secretions or skin-to-skin contact with someone that has a break in their skin. And so I'm like, it's extremely rare. It's not found that often. It is treatable. We do have some antibiotics also used in combination, like tuberculosis, but we have antibiotics out there. Not too worried about this bacteria. And then the last Mycobacterium is the Mycobacterium avium-intracellulare. Most common Mycobacterium infection in AIDS, so even more than tuberculosis and leprae. It's picked up from ingesting contaminated food or water. Problem with this particular bacteria, it's also intracellular, but every cell it gets into, it will kill. And so it will lead to organ failure. So it is a very deadly bacteria. Treatment's becoming difficult, not only because it's a harder to treat bacteria in general because of that Mycobacterium, but also because it's starting to become resistant to the antibiotics as well that are out there. Luckily, not a very common bacteria that we have to worry about either.